All right, so now we're going to talk about the Calvin cycle. So again, we're still in photosynthesis. We're still in the chloroplast. Here is the thylakoids from before. Just kind of scooted over a little bit. So here you can see kind of the edge of one photosystem. You can see the NADP reductase here. You can see the ATP synthase down here. So we have our thylakoids, and then we have our stroma. So remember, this is like kind of like the equivalent of the cytoplasm or cytosol of a cell, but now we are in an organelle that then out here would be the cell. And so the Calvin cycle can be confusing. It can be hard to look at, and it's a lot of steps, and the steps aren't necessarily important. I'm going to go through this to the extent that you can see where the carbons are, how they are being conserved and changed through this process. I do want you to notice that, again, we have NADPH coming from the light reactions, and ATP as well coming from the light reactions into this process. So again, this takes place in the stroma of the chloroplast. And you can see we have carbon dioxide coming in. That's another one of our reactants in photosynthesis. And we have G3P coming out. And then I did put a note here that, remember, this goes on to form C6H12O6 or glucose, so that then we can use that in cell resp. Okay, so there are technically 3 different phases to the Calvin cycle: carbon fixation, reduction, and regeneration of RUBP. So what I really want you to notice is that we have carbon dioxide going in, and we actually will have 3 molecules of carbon dioxide going into this process, and that with each of these 3 molecules of carbon dioxide, you only have 1 carbon. So then if you look over here at RUBP, which is ultimately what our carbon dioxide is going to combine with, RUBP, there would be 3 of them, one in each turn of this cycle. So this whole cycle will happen 3 times. And so each time the molecule of RUBP has 5 carbons. So you take the 5 carbons from the molecule of RUBP, the 1 carbon from the molecule of carbon dioxide, and then you can get this 6 carbon molecule here. We don't even care what that 6 carbon molecule is called, but again, you're going to have 3 of them. So we're conserving. We have 3 molecules of carbon dioxide, 3 of RUBP, 3 of this 6 carbon, and we can trace the carbons through there. So then once you've added these together, you have 3 molecules of a 6 carbon with 6 carbons. So then if you do 6 times 3, you get 18 total carbons. And so remember that you would have 6 in this turn, and then you'd have another 6 in the next turn, and another 6 in the last turn. So this 6 carbon molecule is going to have 1 of them in each turn, but there's 3 turns, so you will have a total of 3. So then this carbon fixation, that's what's going on here. We're taking the carbon from carbon dioxide, we're adding it to RUBP to get another molecule. Then what we're going to do is we're actually going to split that up. So now instead of having 3 molecules of a 6 carbon sugar, we're going to have 6 molecules of a 3 carbon sugar. But if you do the math, again, you will notice we still have 18 carbons. The carbons aren't going anywhere. Then we're going to use ATP, and we're going to use NADPH. And here, all you're using this ATP for is to add that extra phosphate onto the other end. So you still have a 3 carbon molecule, but now it's got 2 phosphates instead of the 1. And so again, our carbons are conserved, but we have to use energy to add that phosphate on. And then here, we're going to use NADPH. It's going to be reduction. Okay, we're going to move the electrons from that NADPH, and we're going to move that hydrogen ion from that NADPH to another molecule. So we're going to reduce another molecule because this NADPH is now going to give that away. And so here again, you see now you have another 3 carbon molecule, and now it's only got 1 phosphate. And so then you end up with your molecule of G3P, but remember we had 6 3-carbon molecules, so again you will have 6, but you have 1 G3P that's going to come out, So 1 3-carbon molecule, and then the other 5 3-carbon molecules are going to go on to regenerate RUBP. And again, you're going to use a little bit of ATP to do this. So when we lost the electrons and the hydrogens from here, we took a phosphate off, we're making some ATP, and now we're going to use it. But so to come back to here, we're going to generate 1 molecule of G3P, and then this will go on to form our sugar, or our C6H12O6. So there's a lot going on here, but the thing to keep in mind is that you're adding carbon dioxide up at the top. You're adding it up here to RUBP, and in adding that one reactant, you then have enough carbon for this whole cycle to happen. And it will happen a total of 3 times. And the idea is you're putting one thing in and you're getting another out. And then this G3P molecule will go on to form glucose, and we can use that glucose as potential energy in the cell.